Hi, Danny. Welcome to the Meet the Brain session. First of all, I would like to ask you about what is your research interest?
My laboratory is working in studying the biology of the neural stem cells, so the stem cells of the brain, and we use these neural stem cells in different aspects or for different strategies. The first one is to try to repair the brain after suffering a damage. For example, the model that I have been mostly used is the ischemic stroke. So after ischemic stroke, the brain is suffering a big damage. Many cells are dying, not only neurons, but other kinds of cells. And our research is focused in trying to repair this damage after this ischemic stroke, using these neural stem cells as a source for transplantation. These cells can be also used to generate in vitro models, so cultures in different ways that can be used for studying the biology of the tissue, for example, the brain in this case, but also to try to study what's wrong when there is a damage, when there is a disease, for example, in the brain. We know, for example, many different neurodegenerative diseases that are affecting the brain, and we can study in the lab with these models that we call brain on chip, the diseases that are affecting the brain.
Oh, looks very exciting. How do you reach your research goals in terms of techniques and methodologies?
We have many different techniques that we use, but we are more interested in the ones that are studying the functionality of the neurons. So how the neurons are working. The neurons are a cell that is sending a message through the cell body, and this message is codified into electric stimulus, and we use different techniques to study these kind of impulses that the nerves are working with. And so the idea is to try to study how the neurons are connected one to each other and how we can improve this connectivity, because this is the main role of the cells and the main function of the brain as an organ. So, for example, we use what we call calcium indicators, that is fluorescent proteins that are able to sense, to feel, and to tell us how is the level of calcium into the cell. These levels of calcium are changing during this impulse, this nerve impulse. And with this, we can study how the neuron is active or not by just looking through the microscope and checking the fluorescence of these indicators.
Mm-hmm. And who are your partners in your research in terms of international or national collaborations?
We have in the lab many different projects. Some of them are international, and in those ones we have partners, for example, from Sweden. That was the place where I was working most of my career. I was doing the postdoc for more than 8 years. But I also have now partners that are in France, in UK, in Israel, or in Switzerland, for example. Mm-hmm. Those projects are very interdisciplinary, so we are working with many different topics, not only with mainly this neural stem cell topic that is our main field of research, but we are dealing with many other researchers that are from the physics, for example. Here in Barcelona we have a very close collaborator in the Faculty of Physics, because to understand how the neurons are working, we also need to know what's the physics under this connectivity, under this connection. So, and these partners in physics are trying to study the neuronal networks, so the cultures or the connections that the neurons are producing. They help us to understand how these networks are constructed.
Mm-hmm. Good. Does your research have a real-world application in terms of innovation and...
Yeah, of course. Nowadays, many different companies are interested in exploiting the use of stem cells from the clinical perspective, but also the use of neurons or neural stem cells to generate models of the brain in vitro is very important, because many different companies are trying, for example, to study different drugs, how these drugs can affect the brain, or you can also, with these in vitro models, you can study how a toxic compound, like a poison, can damage the brain, for example. And since most of the time we were working with animal models, that is very commonly known that these animal models are, with these animal models, the animals are suffering a lot. So it's not ideal, the work with animals. So these in vitro models of human brain allows us to do the same kind of things that we can do with the animals, but only with a piece of culture. And then we are reducing the amount of animals that we use in the research by using these kind of models, for example.
You mentioned Sweden, but how many other countries have you visited during your research career?
Well, actually many countries, because one of the best things of being a researcher is that you're traveling a lot. You're sharing information with many different researchers in the world. And most of the time we have meetings, congress in many different countries. So I was working in Sweden. I was working most of the time also in Spain. But I have been visiting many different countries like US, Australia, Japan, visiting these countries for different meetings or congresses that we are having as a researcher.
Amazing. Well, thank you very much, Danny.
It was a pleasure. Yeah.